All right, today we're going to talk about the Green Revolution. And when we talk about the Green Revolution, we're really talking about 1950s, 1960s. So 65 to 75 years ago now. Um, Agriculture went through a intensification that was driven by trying to increase yields, and in so doing, we went through some technological advancements, including the development of high yielding crop varieties. This required hybridization, which was a key driver of this revolution. We dealt with targeted genetic manipulation. Um, Basically, um, at that point in time, it was driven more by interbreeding of species and interbreeding of specific hybrids of different varieties of, let's say, corn, wheat, rice, and things like that. It was very much a case of we were trying to do a better job of feeding the world. So, our objectives are today are to talk a little bit about hybrid vigor, which is known as heterosis, heterosis and genetic diversity. Hybrids or heterosis is hybrids often outperform their parent varieties due to increased vigor, productivity, and resilience. Basically, they have been bred to have the specific traits of different parents that strengthen the variety and make them a lot more susceptible to being able to grow large yields of whatever crop we are dealing with. Genetic diversity is the combining of different genetic backgrounds to introduce beneficial traits. We start with a very diverse genetic population and then kind of work our way to being very specific about what is going on with the genetics so that we can pick the traits that we want, for lack of a better term, and help in creating greater yields. Um, with plants, this is much easier to do than, let's say, with animals. We can do some animal crossbreeding. Well, when we crossbreed animals, we are genetically crossing um, different varieties of, let's say, cattle and things like that. We are not trying to cross cattle and horses or anything of that nature. Um, the genetic foundations of hybrid varieties, semi-dwarfing genes were introduced in particular India. The introduction of semi-dwarfing genes, the RHT alleles from Norin number 10 wheat and SD1 in rice. In this case, were crossing rice and wheat a little bit. And these genes reduce the plant height, preventing lodging and enabling more efficient nutrient allocation to the grain production. What that really means, I mean, that's a fancy set of terminology and big words, but what it really means is we crossed rice with wheat in an attempt to shorten the stems. I mean, when we talk about dwarfing, we are talking about shortening the stems or the blades of the wheat. And by doing that, more of the nutrients were being pushed to the head or the grain itself. So the wheat grains were getting more of the nutrients and less nutrients were being used up by the wheat stalk itself. In heterosis effect in crops, the hybrid vigor demonstrates the increased yield potential when crossing genetically diverse parents. F1 generation hybrids typically show 20 to 30% yield increases over parent lines, particularly visible in maize crops. And when we talk about maize crops, we were talking about corn. So in the first generation of the hybrids, we are getting 20 to 30% increased yields or more crop than from the parents. From there, when we start getting into F2, F3 generations, we are starting to really increase our yield production. Another thing that we did during the 1950s and 60s was cytoplasmic male sterility, which is a phenomenon where plants are unable to produce viable pollen due to genetic modifications to their cytoplasm, the material outside the nucleus of the cell. This technology revolutionized hybrid seed production, eliminating the need for manual emasculation and removing male reproductive parts from the plant. What does that really consist of? Basically, we created plants that did not have the ability to create male pollen. And by doing that, they could not reproduce themselves. So the pollen was not getting to the ovum of, or the female parts of the plant, and thereby allowed us to control more what was going on with the fertilization of the female portion of the plant. Prior to this, people had to go through and physically remove where the pollen was produced from the plant, and then introduce the pollen that they wanted to introduce. By removing the male portions, now we're preventing that from being a case of allowing reproduction to occur, and we can better control the pollen that was being introduced into the plants, and thereby better control the hybridization of the plants themselves and create the new strains that we wanted to create. Hybridization techniques, conventional crossbreeding. We have interspecific crosses, and that's by crossing different species. For example, wheat and rye, wheat and rice, things like that. Breeders expanded the genetic diversity of crops, introducing new traits and increasing the potential for selecting desirable combinations. By doing this, basically by crossing wheat and rye, we were able to make more sturdy types of wheat. We were able to create grains or be able to, what word am I looking for? Basically, we were able to control. Rye tends to deal with dry conditions a little bit better than wheat. Not that wheat doesn't deal with dry conditions, but rye does it a little bit better. And by crossing with rye, we allowed the wheat to become even more sturdy in desert conditions, and that allowed for obviously more yields in our crops. Back crossing involved the repeating crossing of a hybrid with a parent variety to introduce a specific trait. Basically, what we're doing there is we have plant offsprings of one plant, and we introduce it and cross back with the pollen or vice versa of the parent again. And all we are doing is basically creating more attempts at getting the specific trait that we're trying to find, that we're trying to isolate into a plant, and have that hybrid become a plant species. Basically, we breed for, in the example they use, dwarfism. We keep breeding back to this dwarf plant until basically, and then we grow the plants and we get rid of the ones that are not exhibiting the dwarf trait and breed back to the dwarf plant again. And we just keep redoing this until we've basically wiped out all the tall stem plants and now have a genetic modification to the point that the plants are only producing very small stemmed wheat plants and things like that, which is very important. in dry areas because we don't want a lot of the um, use of the water and nutrients going to producing tall stem plants when we would much rather have the nutrients and water being used to create bigger kernels of wheat. Okay. Then we need to talk about germplasm exchange and global collaboration. The Green Revolution was fueled by global exchange of germplasm or genetic material between research centers. For instance, the transfer of wheat seeds from Mexico to India significantly accelerated the development and dissemination of high yielding varieties. This international collaboration fostered rapid advancements in hybridization and enabled the widespread adoption of improved crop varieties. Basically, wheat that was used to very, very dry conditions in Mexico obviously would work well in India under the same types of conditions. And basically, globalization, or we became international food brokers. The food, we started transporting different plants and even food internationally during this time period and helped with starvation. We still obviously have it throughout the world, but it's much better than it was because of international trade and, for lack of better terms, international education. Education places tend to be a lot more about working together, I guess would be the best way to put it. You got schools like Kansas State University, Oklahoma State University. Um, these schools are agricultural schools, and they work on their own, obviously, but they share an awful lot of information and things like that as well. So they are able to help produce new hybrids, do different genetic testings, things like that. We have moved way beyond just being able to use hybrids by breeding them by normal means. We now have moved to the point where we can do some actual genetic engineering by splicing DNA and things like that. Um, and this is all driven by impact on crop performance. We wanted the yields to increase. We wanted the materials to be more resilient. What do we mean by resilience? Resilience is the ability of a material or a species to withstand bad circumstances. Um, we've created rust resistant wheat, for example. Um, wheat rust is a bacterial fungus type infection that used to really clobber um, wheat harvests. They would create, it would really decrease the yield in the plants and things like that because it would feed off of the plants and cause significant losses in our yield. But by creating plants that are resistant to that, now we don't have to worry about that as much. The yields stay up, the crops stay healthy. That's our big thing. And that ultimately is what led to the dramatic yield increases. Um, for example, Indian wheat production demonstrates hybridization impact with yields soaring from 12 million metric tons in 1965 to over 37 million in the 2020s. So you're looking at, over the course of roughly 60 years, we've increased, India alone has been able to increase its wheat output by 3 times the amount that it was 60 years ago, which is very important when you start considering the population size of India. It's second only to China, and China has gone through several dramatic yield increases as well. This is what has allowed us to prevent starvation. If we were still working with 1960s yields in our crops throughout the world, we'd have a lot of people dying, and the population, the human population would be dying off significantly because we wouldn't have the food resources necessary to feed the world. Some of the limitations and challenges. First one's input dependency. Hybrids often require intensive inputs, which means we have created hybrids that are not necessarily based on just the ability to cope with severe environments. We've also increased hybrids that have a lot of functional demand for fertilizers and things like that. They need lots of nutrients, lots of water, things of that nature. So that's what we're talking about with input dependence. These plants are able to create huge yields, but in turn, they need tremendous amounts of nutrients and water and things like that to create those yields, because they're not creating it from nothing. They are capable of changing water and nutrients into specific food grains and things like that, but they have to have the required materials. It's kind of like baking cookies. If you don't have the right ingredients or enough of the right ingredients, it's going to limit how many cookies you can make. So the more nutrients and things like that you have, the better off you are going to be. Another trait is genetic uniformity. The focus on limited number of high yielding varieties can lead to narrow gene pools, making crops more susceptible to pests and diseases. The southern corn leaf blight epidemic in the 1970s is an example of that. What we're talking about there is just like we've all heard over the years that when you take antibiotics, you got to take your full dose of antibiotics and complete the regime, because if you don't, the infections will become what word am I looking for? Or will become inactive, or the antibiotics will not affect them anymore. They'll strengthen, and the antibiotics will no longer do their jobs. Well, the same thing can happen with plants as well. Different diseases and things like that, different pests can overcome some of the natural defenses that we've bred into plants. And if we are very limited in our genetic setups, that will put us at risk of basically not being able to protect the plants. The world in society has gone to basically banking seeds. Most people don't realize this. It used to be there used to be an island off of the coast of New York that they used to store thousands upon thousands of varieties of every species of grain food in the world, so that they, even seeds that weren't really being used in farming today, just so that they have that backup, so they can go back and, oh, this plant is no longer working because these pests have taken over. We can now breed things to take care of those pests back into the plants with a different variety. Well, that all got moved to the central United States, and they now have that depository at Kansas State University, which is an amazing facility. I lived about an hour and a half from there, so I have seen that facility, and my son actually goes to school there. Farmer reliance, seed saving restrictions associated with F1 hybrids shifted control over seed production to commercial entities, potentially limiting farmers' autonomy and access to affordable seeds. Really, what that means is, in a lot of cases, I have a brother-in-law and a father-in-law that farm almost 3,000 acres, and they used to farm a lot of wheat. They don't as much. They farm more corn now. But back when they were farming wheat, it was sometimes very difficult for them because they had to buy the seeds um, years and years and years ago. They could harvest wheat and take a load of wheat and put it in a grain bin and then take it to a cleaning facility, have it cleaned, and then they could use that for seed for the next year. Well, um, big seed companies have gone to the point now that they kind of neutralize that so that your your grains are no longer, I mean, they'll grow, but they don't grow as well as the seeds that are produced by the company. And that has kind of been hybrid or bred into the plants so that you have to go spend the money to buy new seed every year instead of holding some of your crop back to be able to use that as seed. Oh, direction. All right. So since that point in time, we've had a transition to more modern biotechnology with synthetic biology, gene editing, and marker assisted selection. What that all really means is we are now able to work with the DNA itself. Instead of having to try to breed over and over and over again, we have the ability to understand DNA because of computers and things like that. We have the ability to understand DNA and know where specific traits that we're looking for are found on the DNA, and we can now splice DNA and splice those traits in and start with a very small amount of seed and keep growing those seeds to the point that we now have enough seed to do the hybridization. We don't have to actually go in and wait for that trait to show up and keep trying to rebreed it back in. Now we have spliced that trait into the crop and it's always going to be there. That's what we mean by the synthetic biology, using tools like RNA interference, which is genetic engineering to keep things going. And that's part of the issue with, as I was talking about earlier, with different seeds and things like that. Seed companies can genetically sterilize their seeds so that they will not reproduce if you were to hold part of the crop back and replant it. It's a good money maker for them. I'm not real sure how good it is for the farmer sometimes because they have to spend a tremendous amount of money creating crops anyhow with fertilizer and working the ground and pesticides and buying the plant seed and all that stuff. In some cases, their input costs are greater than their output costs, and they lose money some years just because they don't get the yields that they needed because maybe it didn't rain, things like that. All right. Well, since there's nobody here to ask any questions, I will call this a session and you have a wonderful day. Thank you.